Greetings everyone, Shelbie Raney here. Today I will be conversing with you about a very interesting article titled Cytotoxic Effects of Pseudocerastes persicus Venom and its HPLC fractions on lung cancer cells. Now the purpose of this study was to evaluate the cytotoxicity of P. persicus venom, a type of snake venom, and its protein components on both normal cells and cancer cells. In order to do this though, the researchers performed an in vitro study involving the venom being exposed to A549, which are lung cancer cell lines, and HUO2, which are normal fibroblast cells. Now snake venom itself is an elaborate mixture of organic and inorganic compounds. These compounds can include peptides, enzymes, and low molecular mass proteins. Additionally, some studies have shown that certain snake venom peptides have actually induced apoptosis in cancer cell lines. And some drugs derived from snake venom peptides are currently in preclinical and clinical studies. Now you may be wondering what P. persicus is. Well, it is the scientific name for the Persian horned viper. It is a viper species that is endemic to the Middle East and Asia, and it gets its name for the horn-like structures that are above its eyes as shown in the photo, and for its dispersion in what was once Persia but is now modern-day Iran. The venom itself has hemorrhagic activity and is potentially coagulopathic, meaning that an envenomated prey subject would have impaired blood clotting once exposed to the venom. Besides these two things, there's very little information about the specific activities of this venom. Additionally, there's no anti-serum for this snake venom as it currently stands. Now, for the method section of this article, the researchers were able to prepare the P. persicus venom by dissolving its lyophilized form in distilled water, and they also prepared cell line cultures of A549 and HUO2 in 96 well plates in triplicate form. They then incubated these plates overnight with venom concentrations ranging from 0 micrograms per milliliter to 50 micrograms per milliliter. Additionally, they performed an assay to measure capsase 3 and 9 activities, which is measurement of apoptotic effects in the cells, as well as lactate dehydrogenase release assays, or LDH release assays, which measures necrotic effect in the cells. They also performed venom fractionation by high-performance liquid chromatography, commonly referred to as HPLC. This is important because they were able to obtain 4 different fractions from the crude venom. However, they found that fraction 21 yielded the best results in causing apoptosis in the cancer cell lines. Additionally, they performed morphological studies from the cells incubated with the venom, and they performed data analysis. Upon finding that fraction 21 had the best results for causing cancer apoptosis, the researchers decided to just work on that fraction for the rest of the study. Now, in the results section, here, figure 2, section A, which relates to cell viability of A459 shown in dark gray and HUO2 shown in light gray. As you increase fraction 21 concentration, you're also decreasing cell viability. More importantly, though, you're decreasing the viability of A459 cancer cells, but you still are having some HUO2 decrease in viability, so they perform more tests to find out why. So in section B here, we look at the capsase 3, 9 activities of A459 shown here and HUO2 shown here. As you increase fraction 21 concentration, you do indeed get an increase in these 2 enzyme activities, which in turn relate to increase in apoptosis. Now, when you look at HUO2 here, an increase in fraction 21 does not necessarily mean an increase in capsase 3-9 activities. Very little difference, so great that we're having apoptosis over here in cancer, but there still doesn't account for why there's not that much cell decrease in cell viability in HUO2. So they did another test. In section C here, we looked at lactate dehydrogenase released into the cell media, and we find out why HUO2 was doing bad. As you increase fraction 21 concentration, you're actually increasing the necrosis of the healthy cells of HUO2 when compared to A549. So you would have to use a lower concentration in order to prevent necrosis of the cells, of the healthy cells, while maintaining a increased apoptosis of the cancer cells. After the results of their experiments, the scientists were able to determine that as you increase fraction 21 from 5, 10 to 20 micrograms per milliliter, you actually cause low apoptosis and high necrosis in HUO2 cells, so the healthy cells, and high apoptosis and low necrosis in the A549 cells, so the cancer cells. After going through the concentrations, they found that concentration of 10 micrograms per milliliter of fraction 21 had the most promising result, with it causing 63% cell death in A549 cells when compared to the HUO2 cells. The researchers were also able to determine that an increase in capsase 3-9 activity probably indicated an induced cytotoxic effect caused by the intrinsic apoptotic pathway in the cancer cells. Also, these apoptotic effects were probably caused through mitochondrial dysfunction and cellular stress. So fraction 21 was causing something in the mitochondria of the cell to undergo an issue with its membrane wall. Either the outer or the inner membrane wall was disrupted somehow, and so it caused cellular stress, leading to apoptosis. Now, the results of the study indicated that fraction 21 in lower doses, so 10 micrograms per milliliter, had high cytotoxic activity. So as mentioned before, great for apoptosis. That is what we wanted. However, as we said before, higher doses do cause necrosis in healthy cell lines, and this is because of the high toxicity of fraction 21. In conclusion, fraction 21 at low concentration, so 10 micrograms per milliliter, did display promising anti-cancer properties by inducing apoptosis in the cancer cell lines. However, further investigations really need to be performed in fraction 21 in order to determine its mechanism of action, because we want it to perform the apoptotic effect in the cancer cell line, but we do not want it to cause an increase in LDH, so causing necrosis in the healthy line. So if we can figure out what's causing it to do its method of action, maybe we can improve upon it and use that as an anti-cancer therapy drug. Take-home message is that fraction 21 has the potential for anti-cancer activity, and it may provide a new perspective for drug treatment against cancer. It has a lot of promising effects, just gotta make it stop attacking healthy cells. Get rid of that, we're golden. Here is the works cited page for images used, and that is the end of the presentation. I will now take your questions.